Okay, so today we are going to model what we've been learning about of protein synthesis. Remember, protein synthesis is in two steps. So first we have transcription, where we make mRNA, and then we will have translation, where we actually make our protein. So in order to do this, we asked you to set up some supplies that was sent home in your packets. So you're going to need your cut out strips of DNA. You'll know it's the DNA because you'll see the T instead of a U, and we also have our nucleotides attached, so you should have 4 of those. And then you're going to need your individual nucleotides, so you'll have a uracil, you have your cytosine, your adenine, and your guanine. So those all should be cut out as well. And you're going to need your tRNAs here. And then you should also have one other thing. You should also have your GTP caps as well. So those are all of the materials. As well as the materials, you're going to want your codon sheet to reference. And then along the way, we'll be creating 2 different structures. So you're going to be creating your RNA polymerase, and then you're going to be creating a ribosome. So if you want to pause the video now and set up those structures, go ahead and do so. The key part for the ribosome is that you want your 2 subunits, and then you want space for your mRNA to align in that codon structure we've talked about. So if you want to pause, go ahead. Okay, so what we're going to do is we're going to be modeling transcription and translation. So remember that the first step for transcription is that you want to have your DNA unzipped. And we're going to model that by lining up our DNA strands that we cut out. So you'll notice in your DNA strands you'll have numbers, so we're just going to line the numbers. This will help when we track your work in your final little exercise to make sure you line them up correctly. So we're going to have a 1, a 2, and then we'll see how far we can get with 3 and 4. We might not have enough nucleotides. Okay, so you don't need to tape this together or anything. We're just going to line it up so that we can transcribe. So the first thing we want to make sure we're doing is that we want to have your top box promoter sequence. So remember, that's where you're always going to start transcription. So if you zoom in on this one, you'll see T-A-T-A, and that's where we'll look for the initial gene sequence. That's always what's going to happen. So all right, so after you've found your initiator, you want to start your gene sequence after that. So in this case, we're going to start with this C. And you're going to connect the different nucleotides that correspond to your initial DNA template, like so. So we know that in this case, C bonds with G, we know that T bonds with A bonds with U, etc. So you're going to start building your DNA strand and continue on for the rest of your mRNA. Once you're done with that, you're going to connect them all. Okay, guys, so you can see I have created my mRNA strand using our non-coding DNA template. And I have signified the RNA polymerase doing its job by taping our nucleotides together. So you can see they're sealed together. In order to be ready to be transported outside the nucleus, there's a couple other things that need to happen to this mRNA strand to basically be sealed so it doesn't disassemble. So one, we need to take one of our GTPs and add it as a start cap. So we're going to add that to the beginning of our mRNA strand. And then we need a poly A tail. What we mean by poly A is we just need a ton of adenine. So as I'm moving down my mRNA, you can see I very neatly added 6 extra adenine nucleotides to the end of our mRNA, and that's just going to seal it in so no codes are accidentally read and a fake gene is made. Okay, so sometimes these can be as many as 300 nucleotides long, but that would be a waste of time for us, so we're just going to add in 6. So if you want to do that and pause the video now, you can. Okay, and now that we're back, what we're going to do is actually transcribe our mRNA. I said transcribe, but I mean translate our mRNA. So what we're going to do is we're going to take our ribosome unit and we're going to be moving our mRNA through it 3 at a time in order to start the process. So you're going to want to get your codon sheet in order for this to happen. So you're going to use your codon sheet and you're going to use your tRNA strands, I guess, and find your codons on here and match it to the appropriate anticodon and find the amino acids. Okay, so the key here is that there's a start and a stop codon, right? So you're only going to have one start codon. Remember, it is AUG. AUG. So looking at our codon sheet, you can see AUG is methionine. So we need to look for the beginning of our gene on our mRNA strand. I see that it is right here, AUG. And I'm going to take my first amino acid and connect it to the AUG. I'm going to write MET on my amino acid. I'm going to do this left-handed. Now I'm going to switch hands. Like so. Okay, so this is the process of creating a protein. Now we're just going to move our mRNA strand along. If I can. Doesn't want to. Okay. So I guess the key here I'm noticing is you don't want to overlap. So I have AUG and now I need ACU. So those are the 3 codons. I'm going to pair it up with my anticodon so I don't lose it. And then I want to go over to my codon sheet. I always like to think of this like a secret code. And I'm going to follow the instructions. So it says first base, and we have our first base of A. So I'm going to look for A. And then the second base of C. So remember, I'm looking over here. And then I'm looking for C. So I'm going to follow it down. Okay. So A, C, and then U. And you literally just look for that U. This one's easy. So they're all going to be THR. So that's what I'm going to write. And then eventually you're going to tape these all together just like you did your, just like you did your mRNA, and then you'll have your protein created. Okay, so that is the entire process. You're just going to finish creating your codons for your mRNA strand, and then you're going to send me that code when you're finished. Have fun, guys.